Alrighty, hello everyone. So I tried to get it where you guys could see my face, but it wasn't working with the videos I have to present, so we just have to do it like this. But this is my unit on mitosis and meiosis, and so we would have already had a couple introductory lessons on mitosis. So we will get started today here with our bell ringer. So if everyone could get out a sheet of paper for this question. It is, please write as many reasons that you can think of as to why cell division is important and why it is important to understand. So while you guys are thinking of some things to write for this, you guys can look at this comic I have below. What did the biologist say when his sister dropped a beaker on his foot? Ouch, mitosis. So I'll give you guys a couple minutes to do that, and then we will come back together and read out a couple things that we wrote. Alrighty, so Susie, how about you give us something that you wrote? Perfect. Okay, so she said healing, such as when red blood cells or skin cells wear out and our body goes to divide and it gets replaced for new cells, which is a perfect example. All right, Tony, how about you give us one? Growth, yes? Good one. All right, Cynthia. Single cell reproduction. Yes, and I'm so happy that you said single cell because later in this unit we'll be talking about meiosis. And so that's how we sexually reproduce. And so we'll talk a lot about differences between single cell and multi-cell. And then if someone wants to talk about something about why is it so important to understand, why are we taking all this time to do cell division? Oh, great answer, Catherine. So Catherine said cancer. And this isn't really what I was looking for, but this actually works out really well because we do have a hyperdoc later in this unit that will talk about cancer, which is uncontrolled cell division. So that will actually go perfectly hand in hand with what we're talking about. So I'm happy that you brought that up. So these are great ideas. You guys came up with a lot of great things about growth and healing and reproduction. Um, and then even when stuff doesn't work out, such as cancer. So let's keep this in the back of our mind as we're doing our lesson today. But this was just to help get the ball rolling. So we're going to turn it over to a rap that I found on YouTube. It's kind of silly, but they do bring up some really good points. So we're going to take a look at this.
Susie on the track.
The DNA starts to unwind. The RNA reads the other side. Meiosis is the key to making life. Mitosis copy cells about to die. If my genes go left unread, all my cells are dead. If my genes go left unread, all my cells are dead. If my genes go left unread, all my cells are dead. If my genes go left unread, unwind the DNA. Prep for mitosis happens in the interphase. Then we move to prophase. Spindles start to shape. Chromatids condense as the bubble breaks down. Yeah, I'm talking membrane. Metaphase chromatids meet at the heart. Anaphase chromatids are put apart. See the phase forming a membrane will start. While the cell starts to pinch back apart. Cytokinesis, the cycle is through. That's when cytoplasm is split up into. Meiosis is a lie, except it happens twice. And chromatids are spliced. Please remember the difference is key. Mitosis makes somatic cells. The cells that make up all of me. Except for my sex organs, the haploid cells take half of me. Yeah. The DNA starts to unwind. The RNA reads the other side. Meiosis is the key to making life. Mitosis copy cells about to die. All my my cells are dead. If my genes go left unread, all my cells are dead. Yeah, if my genes go.
Alrighty, so that was kind of fun, but this next animation module that we have here is going to go a bit more in depth. And what this will really do for you guys is give you a visual as it goes through. And I think it's really helpful because you can pause this video and it gives a nice text box underneath explaining what's happening. This will be really helpful when I hand out the assignment we have to do today with a graphic organizer. A lot of these steps are very similar in the way they look, so being able to find those little small differences, which actually will make these huge differences, is going to be really vital for you guys. So we're going to watch this video together, and then you guys can watch it on your own on your iPad. The link it was sent to you guys. So watch it as many times as you need for you to feel like you are pretty comfortable with this. So let's watch this together. His voice is a little, annoying, but it's a really good video. So here we go.
Most eukaryotic cells follow a process of growth and division called the cell cycle. These events include a growth stage, mitosis or nuclear division, and cytokinesis or division of the cytoplasm. Throughout interphase, the cell is engaged in growth and metabolic activities. Interphase can be further broken down into three discrete phases called G1, S, and G2. During the G1, or first growth phase, normal cell functions occur as well as cell growth. In the S phase, DNA replicates, producing 2 copies of each chromosome. During the G2 phase, the cell continues to prepare for mitosis and cell division. Mitosis, or the M phase, has 4 stages: prophase, metaphase, anaphase, and telophase. During prophase, the chromosomes become visible and condense, becoming shorter and thicker. Each identical copy of a single chromosome is called a sister chromatid. The nuclear envelope breaks down and spindle fibers form as microtubules grow out of the centrioles that move to opposite poles of the cell. During metaphase, the double-stranded chromosomes line up along the equator of the cell. The spindle is now fully formed and the microtubules attach to each sister chromatid. Anaphase begins when the sister chromatids of each chromosome begin to separate. The centromere that holds sister chromatids together divides, and the chromosomes move away from each other along its spindle fiber. In telophase, the 2 groups of chromosomes reach the opposite ends of the cell. As a new nuclear envelope starts to form around each group, the chromosomes uncoil and the spindle disappears. The division of the cytoplasm and organelles is called cytokinesis, or the C phase. The result of mitosis and cytokinesis is the formation of 2 genetically identical cells. To summarize the entire cycle, G1, S, and G2 phases occur during interphase while the cell is growing and preparing to divide. Mitosis, or the division of the nucleus, consists of 4 phases: prophase, metaphase, anaphase, and telophase, and in these phases duplicate copies of the chromosomes are split equally. Finally, the cell's cytoplasm divides during cytokinesis and results in the formation of 2 genetically identical daughter cells.
Alrighty. So, like I said before, we are going to fill out a graphic organizer. And what is on here are key events listed on this first column here, and then we also have a diagram or a sketch. And like I said before, it's really important that we know what each stage looks like, because that's going to help us know what we're what's happening. If we can say this is what the chromosomes are doing, this is what the microtubules are doing, that's going to help us with these steps in order. So based on the YouTube video and the module, you guys are going to pair up and fill this out as much as you can right now. And I'll call in a couple people, a couple groups can share what they have for key events, and then whatever we really don't get done right now will just be homework. So yeah, take as much time as you need to look back at those videos and watch that as many times as you need, and then make sure to do some sketches because that is really beneficial to visualize it. So we'll take some time to do that, and then I will call out a couple groups, and then we will reconvene. Okay, so I gave you guys some time. How about Joe and Mason, your guys' group? How about you give me one of the stages and what you wrote for a key event? All right, so you guys are going to do S phase here. Okay, and what did you say for that? Perfect. So Joe said DNA replicates producing 2 copies of each chromosome. That is correct. So that is a great key event that you can put in this box here, and then making sure that you have a picture that somewhat depicts what you said above. All right, how about Sandra and Margaret? What did you guys have besides S over here? Metaphase. Okay, go ahead, Sandra. Yes, that is correct. Did you write anything else down in that box along with chromosomes line up in the middle of the cell? Because that is correct. But there's probably one more key event that we could put for metaphase that you did not include. Yes. Go ahead, Tyler. Yes, the microtubules attached to each of the sister chromatids. Great. So make sure we have both of those for metaphase. Those are 2 important key events, and then making sure that we depict that below with both of those. All right, so let's do one more. All right, how about Chase and Maria, your guys' group? You guys did okay? Yeah, cytokinesis. Perfect, and I'm so happy that you included that last part. So they said division of the cytoplasm, and then they also said it results in 2 genetically identical cells. If that is one thing that you can take away from mitosis, that is the most important thing. 2 genetically identical cells. And we'll talk a lot about these differences when we talk about meiosis as well. So that's great. So make sure you guys have the rest of this filled out along with the diagram below. And this will be really helpful when we get further into this unit as well. So now what I think is going to be another great tool for you guys is a mnemonic device, because there are all these phases: interphase, prophase, metaphase, anaphase, telophase, and cytokinesis. And sometimes it's really easy to get them mixed up. So just for a couple minutes, we are going to think of a acrostic poem or a mnemonic device to help us remember these stages of mitosis. So ironically, I came up with, I placed my anger towards coronavirus. So it's obviously very specific to my situation right now. And you can do this, or you can come up with anything you like, anything that will help you remember it. So this would be interphase, prophase, metaphase, anaphase, telophase, and cytokinesis. So let's take a couple minutes and you guys think of your own poem that will help you throughout this lesson and even throughout this unit plan to remember these specific stages. And then we can share out a couple of them and then come back together and finish up our last thing. All right. Loretta, how about you go and read yours for us? Oh, that's perfect. So she said, I play music at the concert, which is perfect because she's a very great violin player. Um, okay, let's do one more here. Jesse. Okay, he said, I prefer milk and tasty cookies. Great, that's going to help you remember stages of mitosis, then it works for me. All right, so let's put those away and we will finish up with our last thing here. And these are just kind of check for comprehension questions that went along with the video that you guys watched, that little module. This will be your exit slip. So take time to read these 5 questions on the board and just write a letter next to them that correspond to the right answer. They shall be fairly easy if you were paying any attention today during class. So they are the following. Which of the following represents the correct order of the phases of the cell cycle? The division of the cytoplasm is called. Which of the following represents the correct order of the phases of mitosis? Think of your fun poem here. DNA replication occurs in mitosis, and mitosis and cytoplasmic division result in the formation of 2 genetically identical cells. Whoa, it's almost like somebody said that was very important. All right, so take the last couple minutes we have of class and fill out these 5 questions. That will be my formative assessment here. And that'll just be a quick check to make sure we're on the right page for tomorrow's lesson. All right, so good job today. Make sure you finish your graphic organizer if you haven't done so. And we'll be ready to go tomorrow. Alrighty.